Okay, good afternoon and welcome to another IWA Intermittent Water Supply Specialist Group webinar. My name is Razia Farmani. I'm a professor of water engineering from Center for Water Systems, University of Exeter, and I am the current chair of specialist group for this intermittent water supply systems. It's my great pleasure to introduce our speaker today, Dr. Isabel Douterelo. She is a lecturer in water and applied microbiology in the Department of Civil and Structural Engineering at the University of Sheffield, UK. She has over 20 years of experience bringing together multidisciplinary knowledge and microbial research. She has applied her expertise in molecular biology to reveal new bioindicators for eutrophication in rivers, biodegradation processes in wetlands, and the study of aquatic invasive species and the impact of climate change on urban water systems. Her research has further understanding of drinking water distribution systems and the role of microbial biofilms in deterioration of water quality. She's going to give her talk. At the end, we are going to have time for Q&A. Today, we are going to do a hybrid version, as well as having opportunity for you participants to submit their questions in the chat. There will be opportunity for you to turn your camera and microphone on and ask orally rather than typing it. So please either type your questions there, or if you want to raise question by turning your microphone on, that option will be there as well. So thank you, and over to you, Isabel.
Thanks. So I'm going to talk about the impact of intermittent water supply basically in drinking water distribution systems and how the intermittent water supply affects the water quality in these systems. Okay, let me see now. So this is just a slide to give some context of the type of research I'm going to talk about. But we probably, all of you know that currently at least 1/3 of the world population is supplied by intermittent water supply systems. The causes of intermittent water supply systems are different. One of them is mainly water stress, the lack of available water to supply drinking water. Another cause of intermittent water supply can be the losses we have of water through distribution infrastructure, but it can also be a problem of costs related with the use of energy, with trying to pump water constantly all the time. So sometimes it's not just a question of we don't have enough water to supply drinking water. It's a question of it's very costly to keep this system working 24 hours every day. And the practice of intermittent water supply is expected that in the future will increase because we have a population everywhere growing. We have less water, and we need to take into account the effect of climate change in the supply of drinking water. So it is expected that in the future, we will see more systems managed as intermittent water supply systems. I'm going to talk about drinking water distribution systems because I think sometimes they are forgotten. Like most of the efforts are at treatment level, how to treat the water. And there is like an assumption sometimes that the water is going to be okay when it reaches the taps at home or the customer's taps. But that's not the case. The travel after the water, after leaving the treatment, it travels through all these pipes, which are kilometers and kilometers of pipes made of different material. And we have all sorts of microorganisms, as you can see in this picture, that they like to live in these ecosystems, okay, in these drinking water distribution systems. And they don't like to live in the water. They prefer to live, as you can see in the picture, attached to the pipe surfaces, to the inner pipe surfaces, forming biofilms. And most of the research that I'm going to talk today about is about how these biofilms can affect the water quality in intermittent water supply systems. Okay, but as I said, I know that everybody is analyzing water samples and everybody's looking for pathogens in water samples that come out of our taps. But most of these microbes, unfortunately, they live really attached to the infrastructure, to the pipe surfaces. I'm an environmental scientist and a microbiologist, and for me, these distribution networks are ecosystems, okay, and are fascinating ecosystems because they are oligotrophic. Oligotrophic means that there are almost no nutrients for these microbes to survive. Here in the UK, at least, we also have in the pipes a residual of chlorine to keep these microorganisms under control, but they don't care. We are going to find beautiful formations of microbial biofilms in these drinking water networks, creating all sorts of problems. And in Sheffield, we've been looking at these microbes for a long time, and we found all sorts of organisms living there, from fungi, viruses, protozoa, algae, and I can keep enumerating all the type of microbes that we found. So it's a big problem having these biofilms in the water infrastructure, living in our distribution networks. Just briefly, because I'm not sure if all of you know what a biofilm is or what kind of problems these biofilms can give in distribution networks. A biofilm basically are microorganisms that attach to surfaces. They also attach to each other. They love to form these kind of communities, and they are protected against environmental changes, let's say, through what we call EPS, an extracellular polymeric matrix, okay? And if you are a microorganism living in a biofilm, is fantastic, it's perfect, you are protected against different external agents about changes in your environment. But the problem is for water companies or governments around the world, because they create all sorts of problems. They can be reservoirs of pathogens, and they are also reservoirs of antimicrobial resistance. There is a lot of research about how biofilms in pipes are reservoirs of antibiotic resistance microorganisms, and that is a huge public health problem. They can also promote changes in water quality, from odor, changes in taste, discoloration, which in some countries might not be important, but in the UK, the water companies invest a lot of money in solving discoloration problems. And they can also affect the hydrodynamics inside the pipes, and it is well known. The last problem is some microorganisms can corrode materials such as iron in distribution networks. So taking into account all these problems, we don't want, or we try to avoid, or we want to control the formation of biofilms in networks. These are just a few pictures of showing you how the biofilms form. I show these pictures a lot to water companies because sometimes they don't believe me that they have all of these fantastic formation of microorganisms attached to their infrastructure. But basically what happens is you have one of these microorganisms floating in the water. It comes and attach to the pipe surface and start to change that surface and start to produce polysaccharides and change the surface in a way that is attractive for other microorganisms. And as you can see, more and more of them are starting to come, and they start to form this extracellular polymeric matrix, and they grow more and more. And this is a picture of a biofilm that we grew in an experimental facility that we have here in Sheffield, and it's only 28 days old biofilm, okay? So I always say to the water companies here, if this is what you have in 28 days, imagine what you have in pipes that they've been there for years and years. So this was just an introduction of the problem of biofilms in drinking water networks. And you might think, okay, the biofilm is attached, we don't care, it's not gonna give us problems. It's gonna give us problems because if they have dryly conditions within the pipes, like you can see in this picture, changes, the biofilm can be mobilized and can go into the bulk water. So all these problems that I was talking before about pathogens, antibiotic resistance, problems of discoloration, all of that can be favored by the mobilization of these biofilms into the water, okay? And some of the experiments that we did simulating intermittent water supply, most of the problems that we saw related to biofilms is the mobilization of these biofilms when you have events of or periods of intermittent water supply. In terms of how these biofilms can affect water quality when you have intermittent water supply, it's clear that when there are changes in pressure in the system or changes in flow, we can mobilize these biofilms, okay? So the interruption of supply can really disturb this biofilm, leading to problems of water quality that I mentioned before, but also pressure losses in the system can favor the intrusion or biofilms or mobilization of these microorganisms. When you have an intermittent water supply and you empty somehow the pipes, and these pipes already are dry, biofilms are resistant to desiccation, okay? So you still have them there. When you restart the system after X hours of intermittent water supply, again, you are gonna mobilize these biofilms, okay? So it's a big problem. I know that everybody looks at the water, but I think we need to pay a bit of attention of, okay, most of the microbes are attaching the biofilm, what happened with them? So I'm gonna explain you now a set of experiments that we did at the University of Sheffield to try to find out exactly the exchange between these biofilms attached to the pipes and the water. So how do we do that kind of experiments? Because it's difficult to access pipes in the real world where you can simulate intermittent water supply in the UK. So at the University of Sheffield, we have an experimental facility that tried to mimic what happened in a real drinking water distribution system. And it's a huge facility. It's very difficult to take a picture of the whole facility because it's really huge. So this is the picture we have here in Sheffield that better show that facility. But basically, we have 3 different loops of pipe made of high density polyethylene. Okay, that's the material that these pipes are made. And they are connected to 3 different independent tanks. So we can replicate like what happened in each of these 3 independent loops. So we have a loop at the bottom, 1 loop of pipes going around in the middle, and at the top we have another loop of pipe that is going to be connected to the tank. And we use Yorkshire water, okay? We use drinking water from our local supplier to find out what happened with these biofilms that might form in distribution networks. I said, this is the best picture we have because we try and it's impossible. It's so big, the facility, that we cannot really show how big it is, but just to give you an idea. And again, more pictures and a bit of the characteristics of this facility that we use to grow biofilms and to study how these biofilms affect the quality of the water. In this case, during intermittent water supply events. So, as I said before, we have 3 independent loops of pipe. This is the internal diameter. The total length is 203 meters. This goes around. We can control the temperature of the water. We can control the flow. And we can also measure constantly turbidity and chlorine. So we can measure these 2 parameters in real time, okay? But for me, as a microbiologist, the most interesting characteristic or let's say the future of these facilities, the fact that we can put coupons in sections of pipes, and with that coupons that go, so you see these kind of metallic holders, okay, in each of them, we can put one of these little coupons that follows the curvature of the pipe. An interesting thing for me is on the top of these little coupons made of the same material as the facility, we can grow these biofilms, and I can use them to extract DNA and find out what kind of microorganisms I have growing in the biofilms, but the inner part of this coupon, okay, we can take it out and we can look at the microscope. So the pictures I showed you before were just from this little small coupon that goes inside the pipe. So it's a fantastic facility. We can simulate intermittent water supply. We can grow biofilms. We can mobilize them. And I'm going to show you the kind of experiments that we did at the University of Sheffield to simulate intermittent water supplies and then the mobilization of biofilms. I need to say that most of the work I'm going to show you is from Carolina Calero Preciado. She was a PhD student at the University of Sheffield. So most of the slides and the work that I'm going to show you from now are from her PhD. Now she's in Spain in a water company working there. But yes, what I'm showing you is thanks to Carolina. So what we did is we let the biofilms grow in this facility for 60 days. Okay, we wanted a good biofilm growing there, and based on the experience growing biofilm in this facility for a long time, we knew that 60 days was going to give us a very good mature biofilm to play with it afterwards. So we have 3 independent loops running with water, tap water from Sheffield, Yorkshire water, drinking water. So we let these biofilms grow in each of these loops. And then after 60 days, we stop the flow of water. Okay, we were growing these biofilms. You can see here the characteristics at 20 degrees at a low varied flow, which is between 0.2 and 0.5 liters per second. And then we just stop pumping water. And what we did to simulate intermittent water supply is we very, very slowly, because we didn't want to mobilize any biofilm, was to drain these pipes. Okay, as you can see, we use a very, very low flow, 0.06 liters per second. And we left the pipes with no water at different times. 6 hours, which is the minimum time that, for example, in the UK, the water company needs to repair a pipe, sometimes change it, it will take for them to do it. Okay, so 6 hours was like the minimum time that we thought was kind of representative of what happens in real life when a water utility needs to repair a pipe, at least here in the UK. Then we went for 48 hours, 2 days, and based on literature, I know that 2 days is feasible, that in some countries, pipes cannot have water for 2 days. And then we went for the extreme. So we have something on with 6 hours, some pipes 2 days, or one of the loops with no water for 2 days. And then the other extreme will be like leaving this, one of the loops with no water for 6 days. So, okay, we left these pipes with no water. Let's see what happened when you refill the system again. So very slowly, we fill the loops of pipes again. And once we have the pipes full of water again, we play with different pressures and flows to see the mobilization of biofilms. Okay, so what I'm going to show you again is the kind of samples that we took through all these different intermittent water supplies before and after we left the pipes with no water. So during the growth of the biofilm, we were collecting samples, 20 days, 40 days, and 60 days samples of water and biofilm, because we wanted to see how these biofilms were growing in the pipes. Then, when we stopped the system and emptied the system of water, we also took samples. We always analyze the physico-chemistry of the water. We take water samples, but we also take biofilm samples from the coupons I showed you before. And then we restarted the system, but following different stages, okay? First, we slowly return the water, refill the water in the system. Then we increase the pressure. Once the pipes were pressurized, we were playing with increasing the flow and see how that biofilm was mobilized into the bulk water. So here in this graph, I'm giving you a bit of more detail of how these different stages of refilling, basically, the pipes happen. So here you have the flow rate in blue, okay, and how at the beginning, the flow was very, very low. Then we increased the flow in stage 1. We increase more the flow, and we have a final increase of flow. All the characteristics of the different stages in the table below, as you can see, but then we also play with the pressure. Like we have one step of pressure, let's say in stage 0 of this. Let's play and see how these biofilms are going to be mobilized, and then we increase the pressure in the next stages. So playing with combinations of, this is our pressure, let's increase the flow, as I said before, until the maximum. If we keep increasing pressure and flow, the pipes will burst. Okay, so this is like the maximum pressure and flow combination will be a stage 4 that facility let us play with. And these are some of the results from the experiments that I explained you before. So some of them are very curious, and you will see that I think we have some preliminary results that can explain mobilization of biofilms, but I think more needs to be done. But basically here, what you can see is the response of turbidity that we can have, or we had when we restarted the system, okay? In the stage 0, stage 1, stage 2, 3, and 4. So in red again is the pressure, and in blue is the flow. And something curious, I think, because you can see the differences between different intermittent water supplies, is like 6 hours, which is in green here at the bottom, didn't produce a great response of turbidity in the water, okay, which is expected. But the curious thing here is that the 48 hours produce more turbidity when you restarted the system than the 6 days, okay? So this is something that we can discuss, but maybe the biofilms somehow get adapted to these periods, long periods of intermittent water supply. And at a certain point, it's more difficult to mobilize them, but this is what we saw. These are the results that we saw from the pipes. More experiments that we did, and we have help from a colleague in Sejong University. We were trying to find out the characteristics of the organic matter that we have in our system. So we characterize what we call the specific ultraviolet absorbance, okay, what I call SUVA. And this is giving you an idea, if you also measure the amount of dissolved organic matter in your system, of how aromatic, okay, is that organic matter, how difficult are going to be for some microbes to degrade it, but also the potential to form disinfection byproducts. And I guess that most of you know that disinfection byproducts can be carcinogenic. They are a public health concern. So we don't want our system forming hazardous disinfection byproducts. So again, the results I'm going to show you, and I think the ones that are more relevant in this graph, this is the SUVA, okay, that is going to tell us the aromaticity of our organic matter. And as you can see, we have again in green 6 hours, orange 48 hours, and then this, I'm going to call it pinkish or violet, which represents the 6 days, and all the different stages we use to run the system again. And surprise, okay, surprise, because in green, which is the system that was like the loop that was left for 6 hours with no water, we have more aromatic, dissolved organic carbon, more aromaticity, okay, and that means that we probably have a higher risk of formation of disinfection byproducts. So as you can see, some of the results are surprisingly okay. So we have more turbidity at 48 hours, then we have more formation of aromatic compounds at 6 hours. So you need to start wondering what kind of time is better or worse for these processes to take place or to affect the quality of the water. More results, we also were looking at cells. when you restart the system, how many cells are going to pass from the biofilm into the water? And we were also wanted to know if those cells were alive and probably able to create problems, or they were going to be dead and maybe not that problematic, okay? So with the flow cytometer, it's a technique that we use to find out the total cell counts, which is the total cells that we have in the water after we restart the system. And we also look at intact cell counts, which are the cells that are still alive, and they might be able to create problems if people drink it in the taps after you start again the system. So what we saw, and I want to focus again in when you restart the system and you have that kind of flashing effect when you put your system to work again, it looks like in this case 6 hours, as you can see here and here and here. In this case was the system that we kept six days with no water was the one that was giving us more higher counts of cells in the system when you refill and restart the system, okay? And what we saw, and I don't have time to go to a lot of details of all these results, but basically what we saw is the increasing pressure at the beginning was not creating the big mobilization of material, was the actual increase of flow, okay? More results. And in this case, I don't want to go into details because I know that most of you are not probably microbiologists, but I just want to focus in the main findings that we saw from analyzing the DNA and from analyzing all the different kind of microorganisms that live in the biofilm. So we analyze the biofilm that, as you can see here in the graph, we have a column with different colors, and each of those or that color represent a percentage of a certain microorganism in your sample, okay? So the more different colors you have, the more different kind of microorganisms you will have in your sample. And as you can see, we have a lot of them, and at the top you can see bacteria, which are the main inhabitants of pipes. But at the bottom, I also show some results that we have for fungi, because for a reason that we can just guess right now, bacteria and fungi like to live together in drinking water pipes. They seem to collaborate somehow. And it's true that we always find a lot of them when we do experiments here, bacteria and fungi, both of them. But again, we have all sorts of different names that probably if I tell you what they do right now, in 2 minutes, you're gonna forget. So I'm just gonna take you to the results that I think are more important for the management of these systems, okay? So in the case of bacteria, after the intermittent water supply and restarting the systems, we saw that after 6 hours, we have this type of bacteria. They are not very dangerous. After 48 hours, these bacteria were found in the water. But after 6 days, and that's my main worry, we have some mycobacteria and pseudomonas. Some of these species are pathogenic, okay? So the main conclusion for us was the longer you leave the pipes with no water, the higher probability you have that you might find pathogens. The problem we have here, the technique we used was not able to recognize the sequencing of the DNA, was not able to recognize a species level. So I know that I have Mycobacterium, and I know that I have a lot of Pseudomonas, but I don't know if the species that is living there is pathogenic or no. So as I said, there is more research to do. We need to find out more how intermittent water supply really affects public health. But this is a result that we need to take into account, because it's true that it can compromise public health. In terms of fungi, I got good news, because all of these fungi that we found in the water after intermittent water supply, they are mainly pathogens of plants. And only Cladosporium, that was found mainly after 6 days of not having water in the pipe, can compromise human health, okay? But again, I think we need more studies on the implications of microbial ecosystems that grow in the pipe, how that's going to affect public health and the water quality in general. More microbial results are not going to go. Again, I don't think all of you are microbiologists or have a microbiology background, but basically what I'm trying to show in this graph is how, based on the composition of those microbial communities living in the pipes, but also in the water, we have here biofilm. And the ones that we found in the water, how they change. There were changes. Okay, we have again green 6 hours, orange 48 hours. So all of these dots and stars that you can see are samples that we take from biofilm and water. And we saw that with different intermittent water supplies, you have differences in the type of bacteria that were mobilized into the water. So the message here is we need to be careful what we are mobilizing and how long we leave our pipes with no water, because that really affects the type of microorganisms that come out in your tap at the end of the day. And what this graph is also showing us is that there were differences in the bacteria that were released from the pipes with different intermittent water supplies or intermittent water supply times. But the fungi, looks like there are not so many fungi as bacteria, and the fungi didn't show so many differences. They were not, the time of intermittent water supply didn't affect so much the fungi, okay? But again, the message of this graph that I don't want to go in details is the intermittent water supply times affect the type of microbes that are going to be mobilized from the biofilm into the water. And most of those differences were due to bacteria, not to fungi. And basically, this is what I wanted to show you in terms, and to give you an idea of why biofilms growing in distribution system can affect the quality of the water in intermittent water supply. And these are the main conclusions. We did run this experiment, and I think the community working in intermittent water supply would need to do more research, but at least what we saw here in Sheffield is that intermittent water supply were affecting the formation, or potentially affecting the formation of disinfection byproducts, which they are created from the reaction of organic matter with the disinfectant, with chlorine in this case. The pressure, remember that I told you that when we refill the system, we use a combination of first we increase slowly the pressure, but then we were also playing with increases in flow. What we saw is the pressure increases did not really affect the turbidity of the system, and was mainly these increases in flow that were mobilizing most of these biofilm, but not just biofilm. The biofilm can trap, for example, iron, manganese. It's not just the cells contained in the biofilm. It's well known that biofilms can also trap, for example, metals, iron, manganese, and other inorganic compounds. What we also saw is that the turbidity, the results from the turbidity were not related with the results from the flow cytometer when we were counting the total cells and the number of cells that were still alive from these mobilization experiments, which shows that not all the material that is mobilized from the pipes into the water is going to be cells, okay? It's going to be organic. Some of the biofilms, or a lot of cells in the biofilms are active at trapping inorganic such as iron or manganese that can go into the water. And in terms of microbiology, it was a bit scary when we saw that the longer you leave the pipes with no water, the higher percentage of pathogenic, potential pathogenic, because we will need to do more PCR analysis to confirm that those mycobacteria and pseudomonas were pathogenic, but at least the potential for those to appear in your system and in the water after you restart the system is higher. The fungi were there, they were important in the biofilm communities, but they changed less than bacteria. And it was very clear to us that the biofilms living in our system were affecting the quality of the water after you restart the system again. Okay, so these are the main conclusions from this experiment that, as I said, I think the community, all of us working in intermittent water supply, more research needs to be done in the future. And I just want to finish because hopefully there will be some questions after there's some discussion of what I've been explaining. I want to acknowledge Carolina. Most of the work I showed you today, is from her. So thank you, Carolina. Song Kyu, mine from South Korea, he helped us with all these disinfection byproducts, dissolved organic matter measurements. Victor for all the analysis of DNA, and Stuart husband and Joey Boxall for helping us to run experiments with the facility here in Sheffield. And thanks to all of them, I hope I make you think a bit, oh, biofilms, we didn't think about this before, and how they can really affect water quality. And please, if you want to make comments, ask questions, I'm happy to answer. So thank you very much.
Thank you very much, Isabel, for very interesting talk. While we are waiting for the participants to raise their hands or add their question to the chat box, I'm just going to make a couple of announcements. So as you know, we record our webinars and we have a YouTube channel. So if you are interested, you can find all the previous webinars at the IWS Specialist Group YouTube page. Next slide, please. As part of this specialist group, we have guest edited a special issue on intermittent water supply systems. The papers have been finalized, and IWA production team are preparing a virtual special issue for all the accepted papers. This will be published soon, so please keep eye on this special issue. There are a range of papers in this special issue. The papers have been prepared by researchers and practitioners, so hopefully it will provide some reference for ongoing research and practical works that's being done on intermittent water supply systems. Next slide, please. And finally, we are holding regular webinars every month, last Wednesday of every month at 1 p.m. UK time. Our next speaker is Anand Jalakam, and he's going to give his talk on 31st of May at 1 p.m. UK time. Please join us on that again at the next webinar. Also, we would welcome anyone who is interested to give a talk at our webinar series. If you are interested to give a talk, please contact myself or Joe Dalton at email addresses that you can see on the screen, and we can schedule your talk for the future events. Thank you very much. If you have any question, please raise your hand so we can put those to Isabel. Currently, I can't see any.
Or discussion, or somebody thinking that we should
investigate. We are waiting for the participants to ask questions. So I've got one question, Isabel. Did you do any test on the water before entering your rig? So do we know what was the baseline? If the water bringing something with it before entering your experimental facility, or you didn't do that?
Yes, we always take a sample as a control to see what is there, but nothing nothing of major concern. Just my normal microbial communities that live there. The main problem is when you have this biofilm growing and then it's more.
Yeah, very good. And also, how did you clean the pipes before you did the experiment, or you just emptied it and allowed it to sit for 6 hours, 2 days, 6 days, or did you do some cleaning as well?
So we do the cleaning at the beginning of the experiments with very high concentration of chlorine, and then we leave the pipes running for at least 24 hours with very high flow, like to clean them for at least a day, with concentration of chlorine that we need to put it inside and we need to leave the room because it's toxic. That's high. But then for the 60 days, we let the biofilm grow, and then we didn't want to disinfect anything. We wanted to leave the biofilm that grows naturally there, and very slowly we empty the pipes to don't move any biofilm. So the answer is we really disinfected to start the experiments, but after we left the biofilm grow for those 60 days, we didn't want to move it. We didn't want to.
Yeah, of course. Yeah, sure. So basically the pipes were clean and the water that was entering the system was monitored as well. Yeah, very good. And just for my own information, how easy it is to use your facility for quantity measurements as well? Or for example, if a researcher at Exeter University is doing something on hydraulics aspects, not water quality, is it possible to use your facility to do some experiments, or usually you are using for other experiments?
Normally we go biofilms because it's true that these coupons that you put inside are very good for that. But we have visitors from the Netherlands running experiments on where hydraulic experiments, there's no microbiology. So we have people visiting us from other research institutions or universities using the facility to basically do physical experiments, let's say, hydraulics and mainly mobilization of materials inside of sediments or. Yeah.
Okay, very good. Thank you. Sorry, I just saw some hands, but they disappeared. So I'm wondering, Isabel, can you see because you are host? Okay, yeah, please.
Okay, let me see. Two hands rise. Yes.
Yeah, Tang, could you please turn your microphone on and introduce yourself and ask your question?
Hi.
Hello.
This is Xing Tong. Can you hear me well?
Yeah.
Okay.
Yeah.
Yes.
Okay. Thank you. This is a really great talk. I'm really interested. I really appreciate your talk. I actually have two questions about the system. The first one is about the coupon. Does the coupon was set up parallel to the flow, or it's perpendicular to the flow? Because I wasn't really understand.
So it follows the curvature of the pipe. Let me show you. You will see that perfectly fits inside. Let me see if I found it here. Can you see that is curved, it's not flat. So you put it inside the pipe and follows the curvature inside the pipe.
Oh, I see. Okay. So when you sample, is that when you sample the whole coupon?
Yeah.
Okay.
So we take it out and we put a new one.
Oh, okay. I see. Okay. The second question is the second question, when you stop the flow, does the water drain completely, or it's like partially wetted condition? Because why am I asking this question? Because sometimes we will have the water pressurized, but sometimes we will have not pressurized in the evening. And in the daytime, when people using the water again, we pressurize the water again. So during the evening, just to conserve the water, during the evening, the water was not pressurized. It's
Uh-huh.
flowing, but it may not be as high, the pressure may not be as high as the daytime. So I'm curious, would that encourage or increase the biofilm detachment, or that actually doesn't do anything to the biofilm?
So for us was a surprise. That's what I'm saying, that we need more experiments, because we this experiment took a lot of time, a lot of work, and we never repeated. But was strange that the increase in pressure didn't really mobilize anything, was the increase in flow. Where do I have? So this initial increase in pressure didn't really mobilize the biofilm, was the increase in flow. And that was a surprise for us, because you expect that the initial increase in pressure to keep the water flowing in the pipes was going to mobilize. No, it was the flow. I don't know. This is what we saw, and we didn't expect it, to be honest. We expected that this first step here was gonna like flash or move things, but no, it happens afterwards when we started to increase the flow. That was something unexpected, if I'm honest with you. We need more experiments.
Yes.
Yeah.
Thank you. Thank you.
Okay, thank you. And next, Abdul, please introduce yourself and ask your question.
Hi, good afternoon. This is
Hello.
Abdurrahman Shamim, postdoctoral research fellow at University of Exeter. Thank you for the interesting webinar. It was very informative. Actually, my question was also related to the pressure effect on water quality, as you have concluded that pressure didn't have any effect on turbidity and discoloration. So I was kind of wondering if have you also considered pressure transients events by rapidly opening and closing, and whether or not they have some sort of effect on turbidity or not, or you have gradually increased and decreased the pressure. So I was it's very unique, because this could have been otherwise expected, because the pressure transients really affect on water degradation, water quality degradation. So I was kind of curious if pressure transient events have considered in the experimental work. Thank you.
So thanks for your question. We only did the step I showed you before. So we didn't play a lot with pressure transit. Was this change in pressure, and that's it. Another colleagues here at the University of Sheffield play more with pressure transients, but I cannot tell you the results they have. But in this experiment, we only increase it here, and that's it. But I know that pressure transients have an effect, because it's true that other colleagues here in Sheffield, they do that kind of research. I was focused more in what the biofilm is going to do here. Yeah. If you give me your details, I can put you in touch with them if you are interested in knowing more.
Yeah, sure, definitely. Thank you. I would prefer to look forward into that. Thank you.
Okay.
Okay, thank you. And next, Bambas, please.
Hello, can you hear me?
Yes.
Right. Isabel, great. Thank you very much, for your presentation. Quite interesting, very informative. I would like to touch a little bit upon the practical aspect of your research. And perhaps, you can give us some more information on two aspects. If I understood correctly from your presentation, the system was emptied and refilled gradually. Am I right? Did I understand this correct?
Yes.
Yeah, it was not in a way shaken, if I can use that word.
No.
Now, in practice, and all of us that deal with intermittent water supply, I mean, the first thing the operators do is just to open the valves, completely, and then you've got a gush of water, going into the pipes. So, I mean, would that, in your opinion, and that's the first question, would that certainly create a problem with the biofilm, irrespective of the duration of the intermittency?
Yes, because these biofilms are adapted to grow to the hydraulic conditions that they have in the pipe. So if it's an intermittent water supply that is controlled, that always happens at the same time, they are going to get adapted to it. But when you suddenly change the conditions is when you mobilize. And so, yes, you are right. But in this case, we wanted to have the biofilm there just to do the mobilization experiments. Yeah. But you are right. As soon as you change the conditions.
Yeah. Well, so far in practical terms, we've been advising operators to open gently so that the system fills up slowly. But in practice, I don't think it happens. Operators just want to fill up the system as quickly as possible. So that's the first aspect. The second aspect, assuming that we do manage to open the system gradually, so that pressure builds up gradually, we do not have extremely turbulent flows or no turbulent flows as far as we can. What would you say from your experiments would be the optimum intermittency in terms of the biofilm? Because I've seen you checked different durations. What would you say it would be the optimum? I mean, I can guess, but it would be nice to hear your views.
So basically, this was a bit like a jigsaw or a puzzle, because when you look at the formation of disinfection by products, I have the result that 6 hours was bad. So you don't have as much material or pathogens mobilizing that can reach the top, but then you might have a higher risk of formation of disinfection by products. So if you don't have chlorine, which in some countries around the world, they don't even have chlorine, probably this is not going to be a concern, okay? Because they are formed from the interaction of organic matter with your chlorine or your disinfectant. So in terms of the amount of pathogens that can reach and really harm people, 6 days was clear the worst. Yeah. So the longer you leave it, the higher percentage of these microbes that we don't want to have in the water were mobilized from the biofilm. So yes, the longer the worse.
Yeah, the longer the so if we have to have intermittency, then it's best to maintain our duration as reasonably short as possible.
Yeah.
Thank you, Isabel. That's fine. Thank you very much.
Thank you.
Thank you. I don't see any other questions. Isabel, can you see any hands or anything in the chat? I don't see anything.
No.
No. In that case, thank you very much, Isabel, for very interesting, informative talk. And thank you to all our participants for joining us. Looking forward to see you all at next month at our next webinar. And have a nice day. Thank you. Bye.
Thank you.
Thank you. Thank you,
Bye.
Razia. Thank you. Bye.